sequences from different gene elements, or to find the location of specific genes. The technique was developed by E.M. Southern for hybridized probes to DNA fragments that have been separated by gel electrophoresis. The DNA fragments in the gel are transferred to a membrane and denatured. This membrane is then exposed to a labeled probe to allow hybrids to form if the probe is complementary to any of the fragments. The fragments themselves are fixed on the membrane and cannot reform the original double helix. Sites of hybridization can be detected by assaying for a label that has become bond to the membrane. Probes can be labeled with radioactivity or a variety of agents that yield colored products, or even emit light. The hybridization procedure when DNA is in the gel and RNA and DNA probe is called a Southern blot, named for the scientist who developed it. When RNA is in the gel and DNA or RNA is probed, the procedure is called Northern blot. The Western blot, or immunoblot, involves protein in the gel and antibody probe. Sequencing and synthesizing DNA, another technique advanced that changed the study of genetics, was the development of methods for sequencing DNA. In addition, procedures are available to synthesize DNA molecules with defined sequences for use as probes, to aid DNA sequencing, or even to construct entire genes. Two different procedures for DNA sequencing were developed in the mid-1970s. Method and the Sanger D-oxide method, originally developed. Both methods generate DNA fragments that end at each of the 4 bases and that are radioactive. These fragments are then subjected to gel electrophoresis so that molecules with one nucleotide difference in length are separated on the gel. Both procedures involve 4 separate lanes for each sequence determination, one for fragments ending at each of the 4 bases in DNA: adenine, guanine, cytosine, and thymine. The positions of the fragments are located by the autoradiography, and the sequence can be read off the gel. The difference between the two procedures stems from the method used to generate random fragments of the DNA with no nucleotides at the ends. The maximum Gilbert procedure used chemicals that break the DNA preferentially at each of the 4 nucleotides. In the Sanger dioxide procedure, the sequence is actually determined by making a copy of the single-stranded DNA. Using the enzyme DNA polymerase, the enzyme uses deoxyribonucleoside biphosphates as substrates and adds them to a primer. In the incubation mixtures, 4 separate test tubes are small amounts of each of the dideoxide analogs of the deoxyribonucleic triphosphates. Because the dideoxide sugars lack the 3-hydroxyl-contained lengthening of the chain cannot occur. The dideoxide analog thus acts as a specific chain termination reagent. Fragments of variable length are obtained depending on the incubation conditions. The nucleic acid fragments formed are radioactive from using either radioactive primer or radioactive deoxyribonucleic triphosphate. In the reactions, electrophoresis of these fragments is then carried out, and the positions of the radioactive bands are determined by autoradiography by aligning the 4 deoxide nucleotide lanes and noting the vertical position of each fragment relative to its neighbor. The sequence of DNA copy can be read directly from gel. A major advantage of the Sanger method is that it can be used to sequence RNA as well as DNA. To sequence RNA, a single-stranded DNA copy is made, using a RNA as a template, by the enzyme reverse transcriptase. By making the single-stranded DNA in the presence of dideoxynucleotides, various sized DNA fragments are generated suitable for Sanger-type sequencing. From the sequence of DNA, the RNA sequence is deduced by base pairing rules. For determining the DNA sequence of a long molecule such as the whole gene, it is necessary to proceed in stages. First, the DNA is broken into small overlapping fragments, and the sequence of each fragment is determined using the overlaps as a guide. The sequence of the whole molecule can be deduced. The demands of projects that involve sequencing entire genomes have led to the development of automated DNA sequencing systems. With such systems, the sequencing reactions are still based on the dioxide methodology. But fluorescent dye labeled primers or bases are used so the bands can be easily detected. The procedure are separated by automated electrophoresis and the bands detected by fluorescent spectroscopy. In one procedure, each of the 4 different reactions use a different fluorescent label, so that all 4 reactions can be run on a single lane. The results are analyzed by computer and sequence printed out with each of the 4 bases being color-coded. Note that Sanger-type sequence requires a primer of known sequences. That is, a primer must be complementary to a portion of DNA being sequenced. The obstacle is simply overcome because the DNA to be sequenced has been cloned. Therefore, the known sequence to be used will be from the cloning vector. The primer itself can be chemically synthesized. Synthetic DNA. The procedure for making synthetic DNA can be automated so the oligonucleotide of 30 to 35 bases can be easily made in a few hours, and oligonucleotides of well over 100 bases in length can be made if necessary. For the synthesis of longer polynucleotides, the oligonucleotide fragments can be joined enzymatically using DNA ligase. DNA is synthesized in the solid phase procedure, in which the first nucleotide in the chain is fastened to an insoluble porous support, such as silicone gel with particles about 50 microns in size. The overall procedure, the chemical details of which need not concern us here, is shown, and several steps are needed for the addition of each nucleotide. After each step is completed, the reaction mixtures are flushed out of the solid support, and the series of reactions repeated for the addition of the next nucleotide. When the desired length is achieved, the oligonucleotide is removed from the solid phase support and purified to eliminate byproducts and contaminants. Synthetic DNA molecules are widely used for a variety of purposes. In addition to their use as primers for DNA sequencing, they are used as primers for a polymerase chain reaction. In addition, they are used as probes to detect nucleic acid hybridization. Specific DNA sequences we will describe later. How synthetic DNA is also used in a procedure called site-directed mutagenesis to create mutations at specific locations on the genome. Molecular cloning. Molecular cloning is at the base of most genetic engineering procedures and has greatly facilitated the analysis of any genome. The purpose of molecular cloning, also called gene cloning, is to isolate large quantities of species genes of chromosomal fragments in pure form. While it might be theoretically possible to physically isolate pure DNA fragments with a single genes from a restriction enzyme digest of chromosomal DNA, a little reflection will demonstrate the impracticality of such an approach. Consider that even for a genetic simple organism like Escherichia coli, a specific gene represents 1 to 2 kilobase. Out on the genome overall 46,000 kilobase. An average E. coli gene is thus than 0.05% of the total DNA in the cell. The problem is even worse because the coding region of the average genes are not much bigger than in E. coli, but the genome is almost 1,000 times larger. In contrast, the DNA of bacteriophage lambda is only 50 kilobase, and the DNA of some plasmids is less than 5 kilobase, and these genetic elements, a single average gene, constitutes 2 to 40% of the DNA. Thus, the basic strategy of molecular cloning is to move the desired gene or region from a large complex genome to a small simple one. Fortunately, our knowledge of DNA chemistry and enzymology allows us to break and join DNA molecules in vitro. This process is known as in vitro recombination. Restriction enzymes DNA ligase and the synthetic DNA are important tools used for in vitro recombination. Isolation and fragmentation of the source DNA. This can be total genomic DNA from the organism of interest, DNA synthesized from an RNA template by reverse transcriptase, DNA synthesized by a polymerase chain reaction, or even totally synthetic DNA made in vitro. If genomic DNA is in the source, it is generally cut with restriction enzyme to give a mixture of fragments. Joining the DNA fragments to a cloning vector with DNA ligase, the small independently replicating genomic elements used to replicate genes, are known as cloning vectors, and most are derived from the plasmid or viruses. Cloning vectors are generally designed to allow recombination of foreign DNA at a restriction site that cuts the vector in the way that does not affect its replication. If the source DNA and the vector are cut with the same restriction enzyme, joining can be mediated by annealing of the single-stranded regions called the sticky ends. Blunt ends generated by different restriction enzymes can be also joined, and different sticky ends or blunt ends can be joined by the use of synthetic DNA linkers or adapters. The properties of cloning vectors are discussed. Introduction of maintenance and host organism. The recombinant DNA molecule made in the test tube is introduced into the host organism, for example, by DNA transformation. Where it can replicate, transfer of the DNA into the host usually yields a mixture of clones. Some cells contain the desired clone gene, whereas other cells contain other genes generated by joining the source DNA to a vector. Such mixture is known. Such a mixture is known as a DNA library or a gene library because many different clones can be purified from the mixture, each containing different clone DNA segments from the source organism. Making a gene library by cloning random fragments of a genome is called shotgun cloning. Plasmids as cloning vectors. Plasmids replicate independently of the host chromosome. Plasmids replicate independently of the host chromosome. In addition to carrying genes required for their own replication, most plasmids are natural vectors because they often carry other genes that confer important properties on their host. Such genes can be acquired by recombination with the host. In genetic engineering, geneticists add genes to a plasmid in a test tube. Plasmids have very useful properties as cloning vectors.